Okay, so in the last session, I hope I am properly audible, everyone. Okay, so in the last session, we were talking about this topic of components of cell. Okay, we have seen the topics like cell organelle. In that, we have discussed about mitochondria. We have discussed about lysosome, ribosome, Golgi apparatus, endoplasmic reticulum, and then we have also discussed about cell wall. So these things we have discussed. We have even talked about the vacuoles. Okay. So these were the topics which we have discussed in the previous session. Okay. Now, we were starting with this one thing, plastids. So plastids are present only in animal cell or plant cell? Plant cell. Okay. So we have discussed about plastids. Plastids are present in plant cell. In plastids, we have seen that we have got chloroplast, chromoplast, and leucoplast. Chloroplast is for, it contains the chlorophyll. Chlorophyll is required for trapping the sunlight. Then we have seen chromoplast. It is providing different colors to the plants. Why it is providing different color? For the process of pollination. Pollination is basically the process in which pollen grains are transferred from the male reproductive part of flower to the female reproductive plant. Okay. So for this pollination process, we require the chromoplast. They help in attracting the insects, the butterflies, honeybee, etc. So they are all getting attracted because of these chromoplast. Then we have also discussed about this leucoplast. Leucoplast is for storage of food. Basically, when we see that in the plants, there is more and more requirement of storage of food. So in the leaf of the plant, when photosynthesis is being performed, there is a need to store some of the food items for a temporary period of time. So that is being stored in form of our like leucoplast. Then finally, it can be transferred even to the vacuole, or more and more food items can be stored there also. So that is one thing about the different type of cell organelle. Now there is one more thing which is present only in animal cell, okay, and that is centrosome and centriole. In the case of animal cell, there is presence of one thing called as centrosome and centriole. So centrosome is basically the area where centriole is present. Okay. So a section, a region, a section where we see that there is presence of centrioles, they are simply called as centrosome. Okay. So centrioles are elongated pipe-like structure. Centrioles are basically tubular structures, cylindrical structures, which are present in animal cell. What is the role of them? They help in elongation of the cell. One thing is, they are helping in the elongation of the cell. When this cell has to divide, it will help in elongation of the cell. Okay. Centrioles will be going in the different part. It will help in elongating the cell. Plus, it will also help in like breaking these chromosomes. The chromosomes which are there, they have to be like segregated. They have to be also like they have to be converted into two identical parts. So that is being done by the help of centrosome. So it is present only in the plant cell. Only in the animal cell, not in the plant cell. Why not in plant cell? Plant cells have cell wall. We have seen that they are not that much flexible that they can elongate it. Okay. So plant cell also grows. It's not that plant cell will remain of same size. The plant cell will also develop, but it will not be same flexibility. It will not be having the same flexibility as the animal cell. Animal cell can become elongated also. It can just break down into two different cells. But the plant cell will not have the same thing. What will plant cell do instead? In the case of plant cell, what we will see that rather than just the cell getting elongated, there will be development of two different cells inside the plant cell itself. There will be growth of two different parts of the cell wall. They will divide all the parts between them and then they will grow their size. They will increase their size. So plant cells are not going to break and then develop. They will have their internal growth. Internally itself, a cell wall will be created and then they are going to be separated into two parts and then they will grow individually. So that is one thing which we see in case of plant cell. So there is no elongation, there is no stretching of the plant cell. But in case of animal cell, we see that it is elongated and then it is going to form a spindle-like shape. After that, this will form one cell, this will form another cell. So animal cell will get elongated. It will form a dumbbell-like shape like that, and then it will get fragmented into two different parts. So animal cell can get elongated, it can also break down. But in the case of plant cell, it doesn't happen. That's why they don't require the presence of centriole or the centrosome. For your examination purpose, you have to just remember this one thing, that in which cell they are present, in which cell they are not present. So animal cell, they have centrosome. Plant cell, they don't have centrosome. What is the role of centrosome? They help in cell division. As simple as that. Now, these were the organelles which are present in plant and animal cell. We will club them together also and see what is present in plant, what is not present in plant, what is present in animal, what is not present. So if we make a table of this, on one side we have plant, on one side we have animal. So if we talk about plasma membrane, will it be present in plant? Plasma membrane, will it be present in the plant cell? Are you sure? Plant cell? Yes. So it is present in plant. Animal? Okay. If we talk about cell wall, plant cell? Animal? Yes. Then if we talk about centrosome, if we talk about the centrosome, is it present in plant? It is not present in plant. If we talk about plastids, plastids which consist of chloroplast, chromoplast and leucoplast, is it present in plant? Animal? No. Mitochondria, is it present in plant? So mitochondria, Golgi bodies, endoplasmic reticulum, ribosome, lysosome, all of them, they are present in plant also, they are present in animals also. These are all present in plants and animals. If we talk about the vacuoles, in the case of plants, do we have a smaller vacuoles or larger one? So we have seen that in case of plants, we have one large vacuole. We have this one large vacuole. In case of animals, it may be absent. One thing is, it may be absent, or if present, then small and multiple. Small and multiple. It can be found in small amounts plus multiple, small size plus multiple number. Now, when we talk about plant cell and animal cell, tell me one thing, which one is going to be a larger cell, plant or animal? So remember this one part, plant cell is considerably larger one. Animal cell is much smaller. If we look at plant cell, it is hexagonal or rectangular, not always hexagonal, it can be rectangular also. So hexagonal or rectangular, that is the plant cell. When we talk about the animal cell, it is oval shaped. So here it is larger in size. It is larger in size plus rectangular or hexagonal. And this one is oval shaped, smaller. This one is oval shaped or smaller. So that is one difference between this plant and animal cell. There is one more point of difference, and that is about energy storage. That how do they store the energy? In the case of plant, they are storing the energy in form of starch. In the form of starch, they are storing the energy. In case of animals, they are storing the energy in form of glycogen. So here there is one more point of difference, and that is about energy. I am writing the energy part here. You can write it anywhere in your copy. I am writing it here. Starch, glycogen. Starch is basically complex carbohydrate. Glycogen is the simpler form of that same carbohydrate. Okay. So glycogen is the simpler form of carbohydrate. Starch is the complex form of carbohydrate. So plants are storing it in form of a starch, animals in form of glycogen. Okay. So this is one difference between plant and animal cell. So I think by just reading of these functions, I think you are easily able to memorize this. Okay. If you have just gone through the functions of all cell organelles, I think you can easily memorize which of them are going to be there in plant, which of them are going to be in animal. Okay. So this is one general part. Once UPSC has asked about this question, maybe if you are lucky, if UPSC is kind toward you, then they will again ask this. Like there is a general trend that after 5 years, 6 years, 7 years, they are repeating the questions. There is repetition of question. In UPSC exam, there is a repetition of question. Not many, but around 6-7 questions are repeated. Okay. From all the subjects, line by line repetition. I am not saying the normal repetition. Normal repetition can be up to like 15-20. Line by line repetition is also there. Like if you see the question of 2025, you will see one question from medieval India, Arighar, and that was the question in 2016. Okay. So if you see the previous year question, at least you will get help in medieval India. In ancient, you will not get help. In ancient, they invent something new for Buddhism every year. Okay. For Buddhism, there is no end of literature, there is no end of questions for UPSC. If anyone is following Buddhism, they might be knowing less than Buddhism than UPSC. UPSC has more knowledge about Buddhism than what Lord Buddha may be having at one point of time. So they have gone in the deeper part of Buddhism. Okay. Every time they invent something new. And this year, like for next year prelims also, this is going to be a very important topic. Like right now the controversy is going on with respect to Dalai Lama. Okay. So next year also you will find something entirely new about Buddhism. Okay. So if you have to know about Buddhism, don't see the text, see the previous year question of UPSC, you will have greater knowledge about Buddhism. Okay. So if you see the previous year questions of science or environment or that of like history also, will get some repetition. Now, so this is one part. I think plastid we have written already. Chloroplast is chlorophyll having trapping of sunlight, chromoplast attracting the insects for pollination. Okay. So this is one part which we have written. Okay. Now, coming to this one thing, when we talk about classification of cell, we have seen about plant and animal, but there is also one cell called as prokaryotic versus eukaryotic. There is one thing called as prokaryotic and another thing called as eukaryotic. Now, before you write this, let me show you the image here of prokaryotic and eukaryotic. If you look at prokaryotic, this one, the basic cell is our prokaryotic cell. If you look at this, you will find that it has limited features. It might be having a cell wall or it might not be having a cell wall. In some cases, there is presence of cell wall. In some cases, there might be absence also. One thing is fixed, it is going to have plasma membrane. It is also going to have cytoplasm. It is going to have some genetic material. It is going to have at least three things. One is genetic material, then it is going to have the cytoplasm and the plasma membrane. So three basic things are there. Apart from this, you will find that it is having nothing specific in here. No endoplasmic reticulum, no mitochondria, no Golgi bodies, no any other specific feature, no plastids. Okay. So it is having nothing sort of any such developed cell organelle. It was the beginning of the life. It was the time when the life was just evolving. And then we had the first cell in the form of prokaryotic. This was the first cell which came into picture. Later on, what we see that when this cell started to develop, it became the eukaryotic, where we have seen that there is a well developed genetic material in form of nucleus. There is many other things like mitochondria, lysosome, all those things which we have read till now. All the things which we have read like mitochondria, lysosome, endoplasmic reticulum, Golgi bodies, plastids, everything, they are part of eukaryotic cell. In case of prokaryotic, they started with the basics, have a cell membrane or plasma membrane, have a cytoplasm and just the genetic material. Even genetic material is basic level. They are not properly organized. Very small amount of genetic material is there. Okay. So when we talk about a basic level of this prokaryotic cell, you will find that let's make one layer extra, cell wall. There is a cell wall which may be present in this. Okay. Then we have the plasma membrane. Here consider PMS plasma membrane. Okay. Now we have the genetic material. Now you will find that this genetic material is not having any capsule around it or any boundary around it. Its name, prokaryotic. Karyotic means the one which is having a center, the one which is having a nucleus. The one which is not having a well defined nucleus is called as prokaryote. Karyote means the one which is having a center or a nucleus. So prokaryote means before karyote, before nucleus. So this is the stage when nucleus was not well developed. At this stage, nucleus was not well developed. It is having some amount of ribosome. Okay. Ribosome may be present in here. But apart from that, mitochondria is absent or any membrane based, any such membrane based cell organelle is not present in here. Okay. They are the basic cell. Everything happens in the cytoplasm itself. Their respiration, their protein synthesis, all the activities, they are taking place just in the cytoplasm. So it's not that if mitochondria is not there, energy will not be formed. Mitochondria is a specialized powerhouse of the cell, but only in eukaryotic. Without mitochondria also cell can survive. So it's not that at all the time there will be power generation only within mitochondria. Even without mitochondria, a cell can survive. So here we see that like in this all the energy making part, all the protein synthesis, all the transcription, reverse transcription, everything happens within the cytoplasm. It may be having some added features, like if we talk about who are the prokaryotes, any organism which is prokaryotic? There is only one organism which is prokaryotic, the entire bacteria kingdom. There is only one organism, rest all others which are there, they are coming under the eukaryotic section. Bacteria and its predecessors like arc bacteria. Arc bacteria is not even considered true bacteria. So bacteria before bacteria, that is arc bacteria, means even that was not properly developed. So arc bacteria or the bacteria which is present in the contemporary times, that is the only prokaryotic organism. So here you won't have to get confused at all that which one is prokaryotic, which one is eukaryotic. There is only one type of organism having prokaryotic, that is bacteria. Rest all other, from dinosaurs to yeast to mushroom to all others, they are all eukaryotic. So there is only one type of prokaryotic cell, and that is the bacteria. Bacteria or arc bacteria, they are the only. So this is one part. In these cases, now see, many of the bacteria, they are living in water. Many of the bacteria are living in water. Even if they are on the ground, they need something for motility, for their mobility, they need something. In order to even absorb the nutrients, they need some parts or they need something. So what we see that in the case of prokaryotic also, you will find that pili are there or flagella is there. Okay. So pili or flagella, this helps in their motion. This helps in their motion or even catching the prey. If they have to catch something, if they have to get something nutrition, it is helping them. Pili is also something which helps it in absorption of the nutrients or its motion. So they don't have like different things, like for eating we will have like hand, for walking we will have legs. So they have the only one thing, they have pili and flagella. Whatever they have to do with only these two things. They are not having separate legs, mouth, hand, ears, eyes, each and everything is limited. You have pili and flagella, whatever you want to do, you want to move, you have to use this. If you want to eat, you have to do this. If you want to eject some items, you have to do this, use of pili or flagella only. So don't ask me about this, that how they eat or how do they do any other activities. If they have to move, will they use flagella or will they use pili? They will use both of them. Okay. Now in some cases it is written that flagella is used for movement and pili is used for absorption. They use pili also for their movement because flagella is not sufficient enough for their motility. So they will use pili also for their movement and they will use flagella also. So this is one prokaryotic cell having basic features. Okay. Now there is one special thing about them. There is only one thing which is special about them in the present times. Even bacteria is special, but one thing is very special, okay, and that is they have a extra DNA. They have an extra DNA or extra genetic material. This is found only in bacteria or only in prokaryotics. And this extra thing is called as plasmid. This extra thing is called as plasmid. And in the biotechnology, it can also be called as bacteria technology. Okay. Because our entire biotechnology is starting with bacteria. Whenever we are doing gene modification, whenever we are doing gene alteration, it starts from bacteria. The bacteria is helping us in all these things. Like bacteria can help in gene modification, bacteria can help in gene replication. So they are the ones which are helping us in the entire section of biotechnology. We have GM like cotton also, GM maize also, BT brinjal also. We have all the GM crops. They have been extracting the genes from what? Where they have got the genes from? Bacteria. So entire GM crops which you see in the present times, like today there is one article about GM maize, that if you have seen the newspaper, no? Okay. There is one newspaper which comes like, it is written the Hindu or Indian Express. Generally in the morning they throw it outside the gate, okay, or you can get it on their website also. So in that what we see that, like there is one article which is with respect to GM maize. Okay. So the trial of GM maize is going to take place in the Punjab Agricultural University. Like we have the BT cotton also, which is already there in India. We have BT brinjal also, which is not allowed in India. We have DMH 11 also, which is not allowed in India. Basically, apart from BT cotton, nothing is allowed in India in case of GM crops. But all these crops, they are using genes of what? Bacteria. So bacteria is the base of our biotechnology. Later on we will see this one part. Bacteria has taught us the biotechnology. Bacteria has given us this inspiration for biotechnology. Like the entire gene editing is because of bacteria and sometimes because of virus. Okay. So bacteria and virus are the teachers in case of biotechnology. They are telling us like how you can do the gene editing, how you can modify the genes, how you can fight against virus or how you can fight against bacteria. So that's why bacteria is very crucial and even virus is crucial. So in this case, we find a circular DNA which is called as plasmid. It is separate from the original piece of the DNA. I am making the circle here, but there is no circle. Okay. Because there is no boundary across the genetic material. There is no boundary. So that basic cell is what we simply call as prokaryotic. And when we make an advanced cell, like suppose we are making an animal cell, here what we do, we make a nucleus. Entire genetic material is inside it. Then we make one thing called as mitochondria. Then we make also lysosome. Then we also make endoplasmic reticulum. Then we make highly diffused structure in form of Golgi bodies. Then on these itself, we make certain things which are in the form of ribosome. Okay. Near the nucleus itself, we are also having another area which is consisting of centrioles. Then in this part itself, we are having, here it is lysosome, here it is mitochondria. We can have some smaller structures like vacuoles. Okay. So we have the Golgi bodies here, we have the endoplasmic reticulum here, we have the ribosome over here, vacuoles over there, centrosome here, mitochondria, lysosome and multiple such things inside the cell. Okay. If we make an animal cell, we will add further more things. If we make plant cell, if we are making a plant cell, first we will make a layer of the cell wall. We will also make a layer of the cell wall and we will copy this entire item. Plus we will add plastids also. Like here also we will have the nucleus. Here we have a large vacuole. Then we will have endoplasmic reticulum also. We will have the Golgi bodies also. We will have also the presence of mitochondria. We will have the presence of plastids. Then ribosome is already here. So all these things are present in plant cell. So plant and animal cell are complex, advanced cell. But when we talk about the smaller one, when we talk about the prokaryotic, it is having only few things: genetic material, cytoplasm, ribosome, plasma membrane, and sometimes cell wall. Sometimes cell wall plus little bit extension, for example, flagella, pili, these things. Okay. So internally they are having nothing. Internally, if you look at them, apart from ribosome, they are hardly having any structure. So this is the main difference between prokaryotic and eukaryotic. You can write a little bit about them. Give the heading prokaryotic and eukaryotic. In the prokaryotic, write this. In these cells, there is absence of well defined nucleus. In these cells, there is absence of well defined nucleus. In prokaryotes, plasma membrane, cytoplasm and ribosome is present. In prokaryotes, plasma membrane, cytoplasm and ribosome is present. Third point, absence of membrane bound organelle membrane bound organelle like mitochondria, centrosome, plastids, etc. Write about one more point. It may have cell wall, flagella and pili. It may have cell wall, flagella and pili. For motion and nutrition. In this flagella and pili, you can write somewhere that they are for motion and nutrition. You can write one more last point about this, about the prokaryotes. Prokaryotes are consisting of a small circular DNA. Prokaryotes are consisting of a small circular DNA, which is called plasmid. Small circular DNA called plasmid. Let me give you a brief hint about this, how this plasmid will work. Okay. Suppose that this is the DNA structure, and in the middle of this DNA structure, we see that one section, this is our DNA structure or a part of DNA. Let us say that this one section, it is defective. This one section is defective. Okay. We have to replace it. So what we are trying to do, what we do in this section, that we take a plasmid. We cut the plasmid. Then we fill this plasmid with the genes which we want. We fill the plasmid with the genes which we want. Then using the bacteria, we can get multiple series of these genes. We can make multiple such genes from the use of bacteria. And then we can insert it in the body, or we can insert it in the required DNA. This will help to replace this defective part by the original part. So that is one thing. We will see that in detail when we come to this CRISPR-Cas9 restriction enzyme and everything, but how it actually works. But for the time being, you can remember this one part, that plasmid is one thing which we can use for multiplying any specific gene. If we have to increase the amount of a particular gene, if we have to create or add a gene or replace a gene, we have to use the plasmid. So it is like the photocopy machine. If we have to create the photocopy of a gene, we are using the plasmid. Plasmid can be easily modified. This is a very small piece of DNA. So if we have to change this, we can easily do it. Now think about this. If we have to change a bacteria also, like suppose that there is a bacteria of tuberculosis and we have to harm them. So what we have to do? We have to just do a little bit of gene modification in them. We have to just do a small part of gene modification in them, and just by doing a small gene modification, we can change the entire population of bacteria, the forthcoming population. Like any other future generation of bacteria which is being created, we can do it. Okay. In same manner, if mosquitoes are there, okay, mosquitoes are carriers of the various diseases. If we have to harm their population, so what we have to do? They can be living. We have to just change a little bit of gene. By just changing a little bit of gene, we can make the entire population of mosquito as sterile. So then there will be no future generation. So this is also one method which is called as Bolvachia method. It has already been asked in UPSC exam. So Bolvachia method is one such thing in which we are using the gene modification to modify the structure of the genes or modify the genes of the mosquitoes. So their entire future generation will be lost. Like their entire future generation will become sterile and then mosquito will be gone. This is one of the most effective way of controlling the mosquito population. So no more worries about dengue, chikungunya, and all others. Okay. Mosquitoes are spreading from like small malaria to chikungunya to various other problems. So all these can be addressed if we just target their population. So if mosquito is not working, if the Mortin coils are not working or goodnight coils are not working, then in that case we have the option of gene editing. Okay. So bacteria can be more effective than Mortin coil. And see, you are always saying bacteria are not good. How much good they are? Like they help in mosquito control also. They help in increasing the production also. Bacteria are one of the most helpful organism for human beings. Now, write about eukaryotic. In eukaryotic, you can write it is the advanced cell having several membrane bound organelles. It is the advanced cell having several membrane bound organelles. Membrane bound organelle. It consists of a well developed nucleus, with a nuclear membrane. Well developed nucleus with a nuclear membrane. Eukaryotic cells can perform several specialized tasks. eukaryotic cells can perform several specialized tasks. Now, generally in many notes in many books, you will find that a comparison is written that mitochondria is present in eukaryotic, it is not present in prokaryotic. Remember one thing, prokaryotic there is no need of dividing it further. One simple thing, ribosome, cytoplasm, genetic material and plasmid. Apart from this, everything is absent. There is no such membrane bound organelle in prokaryotic. So rather than remembering if mitochondria is there or not, if there is vacuoles or not, remember one thing, it is the most basic cell. When life evolved, it started with prokaryotic. So initially it has only limited things. It has got the limited things. So in that it was simply genetic material, cytoplasm, then we have the ribosome and plus somewhat of flagella, somewhat of pili, only these. There is one extra thing, that is plasmid. Plasmid is found only in prokaryotic. Plasmid is not found in eukaryotic. So if you remember these four things about prokaryotic, then you can easily distinguish whether mitochondria is there or not, whether endoplasmic reticulum is there or not, Golgi bodies is there or not. Remember one thing, they have nothing. They started with some basic things and that is all about prokaryotes. There is only one type of organism having prokaryotic cell, bacteria and their predecessors. Predecessors means like arc bacteria. Okay. Why I am saying predecessors? After the prokaryotic, there was evolution of eukaryotic. So eukaryotic evolved later. After bacteria, there was evolution of the eukaryotic organism and then we have different types. So their predecessors and up to bacteria, they are having this prokaryotic cell. Now in eukaryotic, like we have written membrane bound organelles are present. We have written that well defined nucleus is present. They can be having a specialized task. Which one is larger cell? Prokaryotic or eukaryotic? Eukaryotic. Okay. So there is no need of even comparing the shape and size. It is automatically clear that prokaryotics are very smaller cell. Eukaryotics are well defined and very much complex cell. So this is one part where you can get some questions. Okay. Now we are coming to the main portion from where you might expect questions in exam, and that is stem cell versus somatic cell. Stem cell versus somatic cell. This is one classification which we have to discuss, stem cell versus somatic cell. If we talk about somatic cell, each and every cell which you see in your body, each and every cell which you see in your body, that is somatic cell. Like everything which you are able to see from outside or even from inside, they are mostly somatic cell. Like you have red blood cell, you have white blood cell, you have muscle cell, you have skin cell, nerve cell. All these cells which you know inside the body, they are simply somatic cell. If you look at the example, here we are talking about like human body or even other organisms also. Most of the cells which you see, they are somatic cell. So then what is a stem cell? We will see this part. See, when we talk about human body, just look at this one thing. If we are having RBC, red blood cell, what is their role? Apart from looking red, apart from telling people that blood is red, what is the role of red blood cell? To carry oxygen. Okay. So there is one simple thing, to carry the oxygen and the carbon dioxide also. To carry oxygen and carbon dioxide, that is the role of RBC. If we talk about the white blood cells, what is their role? To fight against diseases, to provide immunity to the body. If we look at the nerve cells are helping in processing, receiving of information, etc., etc. So nerve cell also have a task. If we talk about the muscle cells, like there is muscle cell also, so muscle cells also have a task. They are specialized. Like in the heart also we have the muscles, the cardiac muscles. So cardiac muscles have a simple work, that they work in a rhythm. They work in a rhythm so that there is continuous beating of the heart. So there the muscle cells have different work. The muscle cells in our hand or our legs, they have different work. They help in the movement of the body. Think about this. If we have no muscles, how we may have been walking? Without muscles, if you are walking and all of a sudden you fall down, then what will happen? Some bones will break away and go to other places. Okay. So this is helping in a proper movement of the body. This is helping in the proper movement of the body. Otherwise, we will not be able to move our body in a proper manner. So muscle cells also have that role. From the kidney to the food pipe, there are muscle cells with special function. Okay. Overall, what we see that all these cells, they have some special task. They have certain special task in the body. They are doing some specific role. And in our body, there is not any cell which is just lying idle there. They cannot be simply saying that, okay, we are also here, we may not be doing something, but we are sitting here. Okay. So we see that generally if a cell is there, so they are having certain role, they are having certain responsibility, then only they are present there. So they have some predefined work, predefined role in the body. But yes, there are some cells in our body which are not performing any task. There are certain parts, there are certain cells in our body which are not performing any task on regular basis. They are not performing any task on regular basis. So what we see that there are some cells which has no specialized task, no specialized function or task. They have no such a special role. So why we are having them in body? Like heart cell has its role, nerve cell has its role, kidney has its own work. Every cell in the body is there for some work, some responsibility. But there are some cell of the body having no role at all, having no work at all. Okay. On a regular basis, they are just sitting idle, they are consuming the food and sleeping. Okay. Like sometimes you may have seen in your family also, some people may be saying to you that you are doing no task, you are just sitting here, you are eating the food and sleeping, you are doing nothing for the country, nothing for the family. So in same manner, what we see that there are some cells in body having no task as like. Yeah. So in that situation, why do we have them? They are for emergencies. They are for the emergencies. What type of emergencies? Suppose muscle cell is there and due to any problem, due to any accident, due to any such unfortunate thing, some muscle cell has been destroyed or some skin cell has been destroyed. And in that case, we need to repair that. We need to have quick repair of that skin area. We need to have quick repair of that muscles. So in that case, there are certain specialized cells which can convert into into different type of cell. They can convert into different type of cell. Like one such example, in our bone marrow, we have the stem cells. We have the stem cell. The name of that stem cell is hematopoietic stem cell. This hematopoietic stem cell can convert into RBC also, it can convert into WBC also, it can convert into platelets also. And WBC is not one type of cell. If you see WBC, it is, in WBC there is T cell, there is B cell, there is the like monocytes also. Okay. Then there are neutrophils also, basophils also. So multiple types of WBC are there. Then there is RBC, then there is platelets. So ideally this hematopoietic cell, it has no role. But if there is a lack of RBC, this is going to manufacture it. If there is a lack of WBC, it can manufacture that also. So here it is performing no such specific task, but in case of exigencies, it can make all the different types of cells which are present over there. So this has the ability, this hematopoietic stem cell, this can form WBC also, it can form RBC also, it can form platelets also. So that is the role of stem cell, that in general, in normal condition, no work is being done by them. But in special cases, they can manufacture different type of cells. Just to give you an example, when there is a birth of a child, you know that there is presence of zygote. This zygote is what? It is a cell. This zygote is a cell. From here, there is development of embryo. The cell which is present in embryo is called as embryonic stem cell. The cell which is present here, it is called as embryonic stem cell. Now think about this. What is the embryonic stem cell forming? Embryonic stem cell can make skin also. Embryonic stem cell can make muscles also. It can form RBC also, WBC also, neurons also. Basically all the types of cell in the body is formed just because of embryonic stem cell. From the hairs to all other things, where they have come from, everything has come from this embryonic stem cell. From the head to the toe, each and every cell, whether they are nerve cells, bone cells, muscle cells, red blood cell, neuron, everything is coming from just this embryo. So here there is a cell which can convert into almost all the different type of cell. It is called as embryonic stem cell. So we see that there are certain cells like hematopoietic which is present in the bone marrow. It can make blood. It can make different type of blood cell. There are some stem cells which can make everything, in the body. So suppose if we are having access to an embryonic stem cell, if we are having the access to an embryonic stem cell, can we develop a new human from there or not? We can develop a new human also. Okay. Now think about this. There is a child. Okay. There is a small child of 5 years. Okay. And this child is having problem in the heart. From the beginning itself, the child is born with a heart defect. Okay. So their parents are also there. Like if a child is of 5 years, it means he is also having his parents. Okay. So now there is one possibility that we can form the zygote with the help of the parents. With the help of parents, we can form the zygote. With that zygote, we will get the embryonic stem cell. With the help of embryonic stem cell, can we develop a new heart for the child? Embryonic stem cell can form anything. So can we develop a new heart for the child? So if there is any requirement of an organ replacement, we can use embryonic stem cell. This is the possibility that we can do so. Now come to another side of it. So here if we are going for having a heart, there is one challenge. If we look at this embryo, can it develop into a full-fledged child? We are using this embryo to develop a heart. But if we allow, can this embryo become a full-fledged child? Okay. So now we have to evaluate two things. On one hand, you can have a heart by killing the child, okay, or you can have a child and you can let this child die. Okay. So there are two options. If you are giving him a heart with the help of this embryo, a future child is going to die. If you are allowing this embryo to develop into a child, then this child is going to die because of heart failure. So you can see this, the ethical challenge. Okay. If you go to save one life, there will be death of another one. Okay. So if we are going to use this embryo, there is one challenge. There is this ethical dilemma that who are we going to save? If we are going to save this child, it means we are sacrificing another one for this child. Okay. This is one problem. It means we cannot use the embryo. It is going to be controversial. Many religion, many countries, many societies will not allow it. Many countries, societies or religion will not allow it. So then this is one challenge. So let's talk about other stem cell. Okay. That if we are getting a stem cell from adult body, if we are taking a stem cell from adult body, it can be present in bone marrow, it can be present in muscles, it can be present inside the various internal organs, it can be found in the teeth also, it can be found in the skin also. You can get it from multiple part of body. Finding it is difficult, but in the present times it can be done. So we can extract the cell from here, stem cell from here. It is sometimes called as adult stem cell. There is one stem cell which is called as embryonic stem cell. This one is called as adult stem cell. So if we are using it, there is one challenge. The challenge is it may not necessarily develop a heart. Maybe if we have obtained it from bone marrow, it can make blood. If we have got it from the muscles, it can make muscles. Now when you are making a heart, if you are developing a heart, in that case you require muscles also, you require skin also, you require blood vessels also, you require the very fine like cells also which are required in that for making valve and all other things. So one stem cell cannot manufacture them all. It means we cannot use this adult stem cell because it has a limited potential. Embryo, why we cannot use? Ethical dilemma. In case of embryo, there is an ethical dilemma. If you want to save one, one will die. In case of adult stem cell, we have another challenge. It has a limited capacity. It cannot make different type of organs. Maybe it can make blood, maybe it can make muscles, it can make skin, but not a full-fledged heart. So then we come to the next part. Then we make another cell which is called as IPS cell. Okay. So it is not Indian Police Services. Okay. It's not that if you find any IPS, just extract some cell from there. It is not going to help. Okay. So we develop another type of cell which is called as induced pluripotent stem cell. So we know that in the present times we have the ability to modify the genes of any cell. So what we do, we take an adult stem cell and we go for genetic modification of this. We try to do the genetic modification of this. And if we are going for genetic modification, we can increase its capacity. Like with the help of gene modification, we have made our cotton resistant to infection. We can increase the power of a cell also. So here with the help of genetic modification, we will convert this adult stem cell to near embryonic stem cell, not the full embryonic stem cell. Because the moment you develop a full embryonic stem cell, the ethical dilemma will start again. Okay. If you develop a full embryonic stem cell, then again you will have a challenge that if it is an embryonic stem cell, there can be a life. Okay. So we develop it to a near embryonic stem cell, and that's why it is called as induced pluripotent stem cell. We have increased the capacity of it, that now it can make different type of cells of the body. It can make muscles also, it can make skin also, arteries also, veins also, blood also, and any other thing which we want. So this is the third category of a stem cell which we manufacture with the help of technology. There are two natural. One is the adult stem cell, one is the embryonic stem cell. The third one is technological intervention. Why the technological intervention? Because first one has ethical dilemma, second one has challenges with respect to its capacity. So we increase the capacity but reduce the ethical dilemma. So we have a midway, that is iPS cell. So this is under research right now. It's not that we have been able to do it in a full-fledged manner. In a full-fledged manner, we have not been able to do this. Right now also, we are using this stem cell, the adult stem cell, in some cases. Like suppose if someone has got like if someone is having any such accident and maybe muscles have been lost or skin has been lost, so in that case we can use the adult stem cell to create a skin, to create muscles, and we can transplant it on the person. So right now we are here. We cannot go in this part. There is a limitation. We cannot go in this part. It is legally not allowed. It is legally also not allowed. You cannot interfere with embryo. Okay. Interfering with embryo means killing a life. Okay. So that is one challenge. So here there is no allowance, not only in India, across the world. In the entire world, we don't know about China and North Korea. They don't publish their. Yeah. So they don't publish this. Otherwise, in any democratic nation, it is not allowed. So this is one thing. It is not allowed worldwide. There is a global consensus on this also, that we are not going to use embryo. Adult stem cell, it is on advanced stage. In Delhi, you will find more than hundreds of clinics which are using stem cell therapy. Yeah. Then the next one, iPS cell, it is under research. It is preparing for the exam. Okay. So these are the three different stem cells. So I hope it is clear. Okay. So write a little bit first about stem cell. Sorry, stem cell versus somatic cell. Stem cell versus somatic cell. First about somatic cell. First we are going to write about this somatic cell. In this you can write, these are differentiated. These are differentiated or specialized cell of the body. These are the differentiated or specialized cell of the body. Differentiated or specialized cell of the body. They perform a specific task in the body. They perform a specific task in the body. They can divide they can divide to generate, they can divide to generate identical cells. They can divide to generate identical cell. For example, if we are having a skin cell, the skin cell will give rise to skin cell only. So they can divide and form identical cell. Example, red blood cell, white blood cell, muscle cells, etc. Now we are writing about the second one, that is stem cell. See, remember here one thing. In case of this somatic cell, it's not necessary that every cell will divide and form identical. Like RBC, they don't divide. They grow, they become mature, they die. So they don't divide, they are created again by hematopoietic stem cell. So it's not necessary that all the somatic cell will divide only. They can give rise to identical cell. It is property of most of the somatic cell, but not all of them. Now we are coming to the second part, that is the stem cell. So write about this. These are undifferentiated cells. These are undifferentiated. These are undifferentiated cells having no specific task in the body. These are undifferentiated cells having no specific task in the body. Having no specific task in the body. Next, we will write in this. Stem cells become active. Stem cells become active during adverse condition, when body requires higher amount of higher number of any specialized cell. For example, if the skin has been burnt, then stem cell will divide at a very high pace. Pace means speed, at a very high pace, to replace or fill the skin cells. Now, there are naturally, next point, there are naturally two different types of stem cells. There are naturally two different types of stem cells. There are naturally two different types of stem cell. First one, embryonic stem cell. First one is embryonic stem cell. In this, you can write it has the potential, it has the potential to create all the cells of the body. It has the potential to create all the cells of the body. All the cells of the body. When a child develops, when a child develops, when a child develops, it develops because of the embryonic stem cell. It develops because of embryonic stem cell. It is called as totipotent. It is called as totipotent. It is called as totipotent cell. Here, it is called as totipotent cell. What is the meaning of totipotent? Toti stands for total. Potent means it can divide in all. So totipotent means it can divide in total types of cell, means all type of cell. So it is called as totipotent cell. Next, adult stem cell or adult stem cell or adult stem cell or somatic stem cell. So adult stem cell is also called as somatic stem cell. Adult stem cell or somatic stem cell. In this you can write in the later stage of life, in the later stage of life, the embryonic cell, the embryonic stem cell, in the later stage of life, the embryonic stem cell is not present. In the later stage of life, in the later stage of life, embryonic stem cell is not present. It is replaced by somatic stem cell. It is replaced by somatic stem cell. These stem cells have a limited potential. These stem cells have a limited potential of differentiation. These stem cells have a limited potential of differentiation. They cannot develop in all types of somatic cells. Now after this you can write this one heading, that ethical dilemma versus technical limitation. You can give a subheading, ethical dilemma versus technical limitation. I think in dilemma you have only one L. So ethical dilemma versus technical limitations. In this you can write The embryonic stem cells, the embryonic stem cells, the embryonic stem cells cannot be used for development of new organs, or tissues, as in many culture, religion and society, as in many culture, religion and society, embryo is seen as a sign of life, as in many culture, religion and society, embryo is seen as a sign of life. Hence it raises ethical dilemma. In the use of ESC or embryonic stem cell. Hence it raises ethical dilemma in use of embryonic stem cell. Next point, somatic stem cells cannot differentiate into several types of cells. Hence we cannot use them to develop complex organs such as heart, kidney, liver, etc. Now, after changing the paragraph, or in the next line, you can write this. In this case, the use of genetic engineering, provides new opportunities with the help of genetic engineering. We can modify the somatic stem cells full stop. These modifications can enable somatic stem cell organ. These modified cells are called as These modified stem cells are called as induced pluripotent stem cells. Induced pluripotent stem cells. When you will write the abbreviation of this, you have to use small I. Because if you write capital I think you know what will be the problem. Okay. So in this case, when you are writing the abbreviation of this in exam, use the small I, not the capital I. Okay. These modifications can enable, okay, I will repeat it. These modifications can enable the stem cell. These modifications can enable the stem cell to develop complex organs, to develop complex organs, to develop complex organ. These modified cells are called induced pluripotent stem cell. These modified cells are called induced pluripotent stem cell. So see, so this is one part. If you look at them, on one side we have the somatic cell, then we have somatic stem cell, then we have iPS cell, and then we have embryonic stem cell. This is in the order of increasing potency. Okay. If you look at this, they are called as unipotent. They are called as unipotent, means can form only one type of cell. When we talk about somatic stem cell, they are called as multipotent. So it is automatically understood they can have multiple type of cells. Okay. So here they can form multiple cells. Pluripotent. They are pluripotent. So here we see that more capable than this somatic stem cell. Then the somatic stem cell, and embryonic is called as totipotent, means all type of cell may form all type of cells. So that is one thing about this somatic stem cell, iPS cell, and embryonic stem cell. If we see all three of them, these two, they are also natural. Here also this is natural, this is lab made. This is lab made, means we have modified it in lab. So induced pluripotent stem cell is something which we modify in the lab. The unipotent, multipotent, pluripotent, totipotent. Okay. Potency is their ability to divide into multiple type of cells. In somatic cell, you said they can divide. You give RBC's example, which you later told doesn't divide. Yes. See, in most of the cases, the somatic cells have the ability to divide to give rise to identical cell. Like suppose we have any cell, skin cell, muscle cell, or anything. So they will divide, they will give rise to new cell. But there are certain exceptions. Exceptions is like RBC. RBC doesn't have nucleus, RBC doesn't have mitochondria, RBC doesn't have vacuoles. RBC is only having RBC. Okay. Apart from that, RBC is having nothing. Okay. They are not having vacuoles, they are not having mitochondria, they are not having nucleus. So RBC does nothing. They just carry oxygen and carbon dioxide. So RBC is one exception. RBC is just one exception. I hope this part is clear. Okay. So this is one part of our first chapter, that is cells. Okay. In cell we have seen what is cell, what are the components of cell, then we have seen what is the classification of cell. In classification you can be getting the question. You can be getting question with respect to stem cell, you can be getting with respect to somatic cell, you can get it also with respect to prokaryotic, eukaryotic or others. Yes, in online if you have any question with respect to these you can ask, then we will start the next topic. Totipotent means it can divide in all the different type of cell. Totipotent means these modification can enable the stem cell to develop. Indian Police Services is not there in IPS. It is induced pluripotent stem cell. So don't write it as Indian Police Services cell. Through a stem cell can be stop aging. See, there is a possibility, but it has not been possible till now. There is a possibility that in the future we can modify the cells, we can change them and their capacity can be increased. Their capacity can be increased, but right now it has not been possible. If it would have been possible, then why did Joe Biden retire? Like, he is having all the means, he is having all the options. So if he is retiring, you can understand. Pluripotent means they can have, like, see, if you see this is unipotent. Unipotent is just like one. Okay. Unipotent is just like they can divide in just one type of cell. For example, a skin cell can form only a skin cell. If we come to multipotent means a little bit more power. It can develop into some of the more part, like some of the more types of cell. When we talk about pluripotent, it is much more powerful. Here we will mean that it will have like slightly more power. This somatic stem cell, let us say it is dividing into 5 different type of cell. So pluripotent may be able to divide into 15, 20, 30 these types of different cell. So it is having more power. And then this is the ultimate power, like ultimate potency, totipotent. It means it can develop into all the cells of the body. What about technical limitation? Technical limitation was related to this only, the somatic stem cell, that we have a limited capacity. We cannot develop all the cells with the respect of this multipotent or with the respect of this somatic stem cell. We cannot develop heart, we cannot develop kidney, we cannot develop anything. No, BT cotton is not done using iPS cell. See, if you ask something which we have not discussed, we will not understand that. BT cotton is different thing. Do all human cells have same organelles irrespective of their function? No, there can be somewhat change. थोड़ा हिंदी में भी ठीक है. देखो, just to explain you in a little bit Hindi about these 4 cells. सोमैटिक स्टेम सेल वो सेल है, सॉरी सोमैटिक सेल वो सेल है जो सिर्फ एक प्रकार के सेल को फॉर्म कर सकता है. ओनली वन टाइप ऑफ सेल, सिर्फ एक प्रकार का सेल फॉर्म कर सकता है, दैट इज सोमैटिक सेल, इसीलिए इसको हम लोग यूनिपोटेंट बोलते हैं। सेकेंड पार्ट सोमैटिक स्टेम सेल, इसे हम मल्टीपोटेंट बोलते हैं क्योंकि एक से अधिक प्रकार के सेल फॉर्म कर सकता है। लाइक इट इज फॉर्मिंग फॉर एग्जांपल हेमेटोपोटिक स्टेम सेल, ये आरबीसी भी बना रहा है, डब्ल्यूबीसी भी बना रहा है, प्लेटलेट्स भी बना रहा है, तो दैट इज व्हाई इट इज कॉल्ड एज सोमैटिक स्टेम सेल। अब देखो नेचुरली देखें तो वी हैव जस्ट दिस एम्ब्रियोनिक स्टेम सेल। एम्ब्रियोनिक स्टेम सेल जो एम्ब्रियो में मिलता है, एम्ब्रियो इज द पार्ट विच इज फॉर्म्ड आफ्टर जाइगोट, तो इस एम्ब्रियोनिक स्टेम सेल में टोटीपोटेंसी होती है। टोटीपोटेंसी मींस ये सभी प्रकार के सेल को बना सकता है, ऑल टाइप्स ऑफ सेल। जैसे कि अगर एम्ब्रियो है तो एम्ब्रियोनिक स्टेम सेल सिर के बाल से लेकर पैर तक सभी चीजों का निर्माण कर सकता है। दैट इज एज सिंपल एज दैट, फ्रॉम द हेड टू द टो ऑल द थिंग्स कैन बी फॉर्म्ड विथ हेल्प ऑफ दिस एम्ब्रियोनिक स्टेम सेल। और क्योंकि हम इसका यूज नहीं कर सकते हैं, बिकॉज इफ वी आर यूजिंग एम्ब्रियो, तो देयर इज अ चैलेंज कि अगर हम लोग एम्ब्रियो का इस्तेमाल करते हैं तो यू हैव वन ऑप्शन, ईदर यू कैन हैव अ हार्ट और यू कैन हैव अ लाइव चाइल्ड। या तो एक बच्चे को आप हार्ट देकर उसकी जिंदगी बचा सकते हो, लेकिन अगर आपने एम्ब्रियो का इस्तेमाल किया तो एम्ब्रियो एक नए बच्चे के रूप में भी डेवलप हो सकता था। तो ईदर इन एनी वे यू आर गोइंग टू लूज वन लाइफ, या तो एम्ब्रियो को किल करो या फिर यू कैन किल द वन पर्सन हैविंग डिफेक्ट इन हार्ट। ये एक पार्ट है जो एम्ब्रियो में हमें चैलेंज दिखता है। तो दैट्स व्हाई व्हाट वी डिड कि वी हैव मॉडिफाइड द सोमैटिक स्टेम सेल। फिर हम लोगों ने सोमैटिक स्टेम सेल को थोड़ा मॉडिफाई किया यूजिंग द जेनेटिक इंजीनियरिंग। तो व्हाट वी डिड हियर कि वी इंक्रीज्ड इट्स पावर एंड वी चेंज इट इन फॉर्म ऑफ आईपीएस सेल। इसके पावर को थोड़ा इंक्रीज किया, इसको हम लोगों ने इंड्यूस किया। इंड्यूस मीन्स टू इंक्रीज इट्स पावर और टू इंड्यूस सम क्वालिटीज, कुछ क्वालिटीज को बढ़ाना। हिंदी में बोलते हैं इसको प्रेरणा देना, ठीक है। तो इंस्पायर और इंड्यूस सम क्वालिटीज इन इट, ठीक है। तो यहाँ पे जो एग्जिस्टिंग सोमैटिक स्टेम सेल थे उनको इंड्यूस किया गया सो दैट दे कैन हैव मोर पावर एंड देन दे कैन डेवलप कॉम्प्लेक्स ऑर्गन। तो दैट इज इंड्यूस्ड प्लूरिपोटेंट स्टेम सेल। तो आई थिंक अब ये चारों समझ में आ गया होगा। हाँ, स्टेम सेल अम्बिलिकल कॉर्ड में प्रेजेंट हो सकता है, यस, it can be present in the umbilical cord. At that time it is present in umbilical cord, but it is not the embryonic stem cell as such. Embryonic stem cell is mostly there when the child is getting developed. In the initial stage there can be presence of embryonic stem cell. Later on, at the time of this delivery of the child, embryonic stem cell may not be easily found. Okay, so this is one part which we have discussed. Now after this we have to discuss the second section. Yes, nahi, having a twin is entirely different process. Twin is something which is random selection. Twin cannot be decided on the basis of like technology as of now. If anyone is trying to have twin, that is a totally random case, that suppose there has been two eggs which has been released by the fallopian tube at the same time, so then in that special case there can be a twin. Like what generally happens that in the case of female there are a pair of ovaries. Each month one ovary is releasing one ovum, one egg cell. Now in random case, if there is a possibility that in one particular month when the couples were trying to have a kid, at that time if both of ovaries have released one ovum cell each, and sperm cells are already in millions of number, so if there is two egg cells which has been released by both of the ovaries, like one each, in that special case there can be formation of twins. So twins is formed when both of the ovaries are giving the egg cells. So that is quite random. Even if we are doing it in lab, like when we are doing it in lab also, so then we are taking one cell, one ovum cell. In labs, if we want, we can have two different ovum, then we can mix it with the sperm and then we can create two kids, or we can create any number of kids. But naturally the twins which are getting formed, it is of a random process. It may happen, it may not happen. So we cannot decide it. No. Embryonic stem cell is not like causing this development. If there is a fault in the process, if there is a fault in pregnancy, you may have seen that many a times the child is not developing properly. Sometimes they are having two heads or more than that. So that is a special case. So that is not twin. Twin is formed when two egg cells are released simultaneously. So that is a problem in the birth process where there has not been proper development of the child, or where there has been any problem in the process. It may have gone extra, it was not properly regulated, then it may happen. Having extra organ also reflects that there may have been some mutation in the genes. If there have been some mutation in the gene, then can be a person may be having like two body parts in the top or at the bottom. So that is one different thing. It is not twin. It is not twin, it is a different case. In some cases, sometimes twins are joined together. So that is also a special case, that two twins are developing, they join together. That is one special case, but it has nothing to lead with like embryonic stem cell. Two twins have been formed, plus there is problem in the pregnancy period, then it becomes. No, technology is not there for, we cannot even decide whether it will be male or female. Deciding twins is a different game altogether. So we cannot even decide whether it will be male or female, it cannot be even predicted. So that is totally 50-50. Last few years back, one of the company started a product, and they said it openly that if you are consuming this product, you are going to have a male child. So then there was a case by ICMR in the Supreme Court. One of the company, like that company has been there in the news continuously. It is an Ayurveda based company. So they have launched a product and they have said that you can have a male child if you are having continuous consumption of a particular churn. So then after there was a case in Supreme Court also. There was a case filed by ICMR itself. And they said that how it is possible. Where we have missed. We have been searching for so long and we have not got anything. Then there was a demand of public apology by Supreme Court. Supreme Court has said that you have to publicly apologize for this. In the entire newspapers throughout the country, they have given a written apology. So in last year, perhaps in 2024 or 23, this case was there. So it cannot be done. Not taking the name of company, but you know. Now we are coming to the second part, that is animal kingdom classification or kingdom classification. Yes, same company has also found the cure of COVID also. Now, so here we have this one thing, a smaller topic, which is about kingdom classification. Why we have started this? Let us try to understand this first. We have seen that in the cell topic, we have studied two things. One is plant cell, one is animal cell. So we have classified the entire world initially in just two things, that either there is a plant or there is an animal. So initially the entire organism classification was on just two bases, that either something can be plant or something can be animal. But later on it was seen that it's not like that. Every organism which is resembling in this cell structure is not necessarily a plant or an animal. Some animal cells may show the property of plant, some plant cells may show the property of animals also, or some having plant cells characteristics may be animal type also. So due to this, finally what we see that multiple classification started, multiple classification with respect to organism. And one such classification was given by R.H. Whittaker. R.H. Whittaker. He provided a 5-kingdom classification. He provided for a 5-kingdom classification. One of them was Monera. Then we have another one in form of Protista. Then we have the kingdom Fungi. We have the kingdom Plantae and Animalia. So 5-kingdom classification was provided: kingdom Fungi or kingdom Fungi, Monera, Protista, Plantae, Animalia. These 5 classifications were said. It was said that not everything is plant, not everything is just animal. There can be different classification. So we have this first group, which was the basic group, Monera. We will try to understand like what are these 5 different groups. First of all, I will explain it to you right now. We will write about it later. So let me give you an overview of these 5. In the first group, we are having the most primitive organisms. Primitive in the terms of evolution. In the terms of evolution, not in terms of their capacity. They are very much powerful organism. So they are primitive in terms of their evolution, like the prokaryotic cells. The entire prokaryotic organisms are just here in Monera kingdom. All the prokaryotics which are there, they are all lying here in the Monera kingdom. Now who all are there in Monera then? If prokaryotics are there, so which organism we are talking about? Bacteria. So all the bacteria which is there, they have their own kingdom. The bacteria have their own kingdom, kingdom Monera. Here you can also count their predecessors, archaebacteria, but it is basically the bacteria only. So they have got their own kingdom. They are unicellular organism having the prokaryotic cells. So then we have some other also. We have some other unicellular organism also. We have like Plasmodium is there, Paramecium is there, diatoms is there, dinoflagellates is there. There are so many other unicellular organisms. So any unicellular organism which is not bacteria, any unicellular organism which is not bacteria comes in the second category, which is Protista. Means all other like amoeba and everything, they are all coming under this second category, that is Protista. Here they would have prokaryotic cell or eukaryotic. In Protista, they are going to have prokaryotic or eukaryotic. Remember one thing, prokaryotic is just here. Apart from that, there is no prokaryotic. So prokaryotic is only there in the Monera kingdom. Rest all other kingdoms will just have the eukaryotic. So all the unicellular organisms which were having eukaryotic cells, they were placed under this category of Protista. Then we have got all unicellular combined in these two. They are the category of unicellular organism. They are all unicellular. Unicellular means one single cell. Single cellular organism. Bacteria has their own kingdom. All other unicellular combined together come in Protista. But then there was another microorganism which was not so micro, means a little bit. It was not fully plant, it was not fully animal, but it was not fully microorganism. So there was another organism which got its own kingdom, and that kingdom belonged to fungi. So fungi is not a plant. Fungi is not a plant. Mushroom is not a plant. Mushroom is an organism. So all those who consume mushroom, you are not vegetarian. Okay. Because vegetarian concept is for plants. Okay. That if you are using plant-based products, then you are vegetarian. So if you have used mushroom anytime, That is a willingness. Like you may say that everyone is using bacteria, everyone is eating bacteria. This moment also you are eating some bacteria. Okay. So if we sideline the bacteria also, many people willingly eat mushroom. Okay. So if you eat mushroom, mushroom is not a plant. Mushroom is an organism. Like the cockroach, it is also an organism. Okay. So it is an organism which comes under this fungi category. Okay. So here what we see that they have got their entire kingdom, that is fungi. Okay. Then all the plants of the world, they were classified in this one, Plantae. The plants which were there, we will see them in detail. Right now we are just seeing the overview. The plants have come here in the kingdom of Plantae. So plants make their own food. Do all the plants make their own food? Which plant is not making its own food? Insectivorous. So how do they get the food? They also make their own food. Insectivorous plants also perform photosynthesis. All the plants which are there, they are making their own food. Even if they are insectivorous, they are also making their own food. So plants make their own food. Now, why do they feed upon the insects? Yes, they need minerals. The soil in which they are developing may not have nitrogen, may not have phosphate, sulfate or other things. So that's why they require nutrients and they can get it from the insects. So that's why they are consuming the insects. Otherwise, they are doing photosynthesis. Okay. So they do this photosynthesis part and then they eat the insects also for their nutrient intake. They are not eating them as per se. They are just killing them, deriving the nutrients from them and then using that nutrient to perform photosynthesis, make their own food. Okay. So all the plants which are there, they are making their own food. They can be insectivorous, but even then they will make their own food. Then comes the one final kingdom, which is Animalia. Okay. So Animalia is all of us. Okay. All of us, like everything which you see, spiders, rat, cat, dog, elephant, humans. So we all have been classified together. See, we have not been kept together with any other plant or anything. We are in the category of Animalia. So we are in the same category as mosquitoes, as cockroaches, rats, frogs, everything. So whenever you are finding these things not so attractive, so think about this. They are from the same category as we are. Okay. So they are also from the same kingdom as we are here. All these organisms are coming from the Animalia section. In which category we will place virus? Will it be Monera or Protista? It will not be there. It is of living organism. It is of living organism. So remember one thing, again one reminder, viruses are not living organism. Okay. We have seen it in the past also. Viruses are not living organism. They will not be part of this group. Okay. So five kingdom classification is just for living organism. So we will see that entire thing in detail in the next session, that is tomorrow. We will see about insectivorous also, plus this is a small topic, so after that we will start with human body. Okay. We will see this human body also, like what brain we have, okay. What is the role of the brain and everything, so we will talk about that. Okay. Just wait, we have some questions. Okay. So if there is any other question from online section, I think we have already discussed questions. Like symbiosis relation, so algae and fungi is different thing. Lichens, we will read this. Any question from online section? What is process of induced pluripotent cell? Please a little bit. See, the class is not over. I am going to give some questions. There is one question. See, there is one question that what is the process of IPS? In the process of IPS, what we do? In the process of IPS, what we do? First we extract this somatic stem cell from the body. Then we modify the genetic structure of it. Okay. And as per our requirement, we can make it like pluripotent. Whatever cells we want to form, we can train it like that, or we can modify it like that. Plasmid, yes, plasmid is found only in bacteria, only in prokaryotic. There is one question, can this not happen that if someone is going for abortion, then embryonic stem cell may be used? We can do it, but if someone is going for abortion also, so that is not even preferred by many of the religion, culture and society. So then it is very difficult thing. People will say this, that if you are going to use the embryo, why not save that baby? People can say this also, that suppose someone is going to go for abortion and scientific community has taken that embryo. So then there will be a question from scientific community that you are preferring one's health over a new baby. So rather than saving one person, if you are so much kind, use that embryo and develop into a new child. So there is always this ethical dilemma. अच्छा, plastid का नहीं लिखाया था. Okay, if I have not dictated that in the beginning of the class tomorrow, I will dictate it. We can make, like with the help of induced pluripotent stem cell, we would be able to make heart or any other complex organ. No, it is not possible, Ritesh. It is not possible. I have explained that gender identity cannot be done before the birth of the child. It is illegal. It is not about the technical capacity. If we talk about the technical capacity, it can be estimated in the first ultrasound itself, but it is not legally allowed. By one embryo cell, we can divide it or not. One embryo cell gives rise to the entire 6-foot human. The entire 6-foot human is made of just one embryo cell. Neuron cells do not divide. It is not about neuron stem cell. Neuron cells also don't divide. After a certain period of time, they don't divide. So when we will talk about human brain, we will see that if neurons start dividing, you will get another excuse for not revising the topics. If neuron starts dividing, then you will not remember anything. Then you will tell me next day that we don't remember anything about cell. Our neurons have divided. Yeah. So if neurons start to divide, you will lose all the memory. We can make each and every organ. Technically, it is possible. But right now, there is a limitation that in many countries, it is not allowed. For some of the things like for developing the stems, for developing the skin, for developing the muscles, it is being done. It means ESC simply means seed which can make a new plant. That is the same thing. Doctor can identify, they will not tell you. Why are you worried about the child right now? There is still a lot of time. Don't worry about the child right now. Still there is a lot of time. Maybe by then there will be some change in the law. If you are not planning immediately, then you don't have to worry about it. Okay, so that will be all for today. Tomorrow we will start with this classification of organism, and then we will see human body also. Okay. Any question from offline?